Good morning, everyone. It's a pleasure to update you on the progress we've made at Maddie and share with you some new clinical data that we've generated. Now, we're developing the Evolute, which is a punctal plug-based drug delivery technology. We think that this can efficiently deliver drug to the tear film. And one of the ways we accomplish that is we have a solid drug-eluting core. We wrap it in a membrane and then glue it at one end, marry it with a punctal plug so that the tear really pulls the drug out of solution and only can go into the tear film and not into the surrounding tissue. Since we're talking about a punctal plug, it's important to highlight retention rates. These are retention rates from 2 multi-center U.S. studies showing about a 94% plug retention rate over a 12-week period of time. So then the question becomes, how broadly can you use this technology? And we're developing it and highlighting the programs that we have that are active across all of the 4 major disease segments of the anterior segment. We've done most of our clinical work in glaucoma. We've also done clinical studies in olopatadine. I'm going to share with you new clinical data today with Nepafenac, an anti-inflammatory, and we're nearing completion of a formulation of cyclosporin for dry eye. So this technology can be used broadly across all the major disease segments. So I presented about 40 patients worth of data, interim analysis data from this study, which is a phase 2 study looking at Nepafenac Evolute in patients undergoing cataract surgery. It's a double-masked study, placebo-controlled. So every patient got a plug placed in their eye. Plugs were placed 1 to 2 days preoperatively. And we were looking at pain and inflammation after cataract surgery, and the randomization was 2 to 1 in favor of the active. The full data results will be presented by Dr. Eric Donenfeld at the main meeting on Monday. And this is full data set with 63 patients. Now, the study wasn't powered to show statistical significance. Baseline demographics were balanced in terms of age, gender, and race, and also in terms of the surgical procedure. Important to highlight, since the plugs were placed preoperatively, that we only had 1 plug dislodged following the lid speculum placement and all the lid manipulation that goes on from cataract surgery. So retention rates were excellent in this study. Let's turn to some of the data. This is patients percentage of patients with no pain at the day 3 post-op, about 75% in the treatment arm and 38% in placebo. This was statistically significant. This is probably the most interesting data point in the study. This is percent of patients with no pain throughout the entire post-op period, and we measured patients at day 1, day 3, day 7, and day 14. 42% of patients in the treatment arm experienced no pain after cataract surgery versus 0 in the placebo arm, and this was highly statistically significant at the less than 0.001 level. Turning to inflammation, this is percent of patients with none or trace anterior chamber cells at the day 3 post-op visit, about 80% in the placebo arm versus 63%. This was trending towards significance but didn't quite achieve it. But when we expand the criteria and look at none, trace, or mild anterior chamber cells, again at day 3 post-op, we did achieve statistical significance with 97% of patients in the active arm versus about 65% of patients in the placebo arm. When we look at another measure of inflammation, anterior chamber flare, patients with none or trace, we approach significance with this, about a 20–25% difference in anterior chamber flare. And then when we look at visual acuity outcomes, these were originally collected as Logmar, but converted to Snellen at 2032, mean visual acuity at the day 14 post-op visit for the treatment arm versus 2050 for the placebo arm. This was statistically significant as well. Another measure of visual acuity is uncorrected visual acuity. This is percent of patients who are 20/25 or better. This was also statistically significant and about a 40% difference between the active and placebo arms. And then finally, best corrected visual acuity, percentage of patients who are 20/20 or better, again, about a 30 to 40% difference between the active and the placebo arms. So across this study, we showed statistical significance in pain, inflammation, and better visual acuity, which highlights why we think this is an efficient way to deliver drugs to the surface of the eye. In terms of safety, a total of 14 adverse events were reported, 19% in the Nepafenac arm, 29% in the placebo arm, and no serious or unexpected adverse events. Upcoming milestones, we plan on initiating a travoprost glaucoma study, another cataract study with dexamethasone. We'll complete our formulation of cyclosporin, complete the manufacturing scale-up to enable us to start a phase 3 study next year, and then complete the formulation of olopatadine for allergy. This platform is well protected with 114 global patents that have issued. We're still on a run rate of getting a patent issued approximately every month. So in summary, there is a wide range of compounds that we've already formulated, more that we would like to formulate. This is a very flexible drug delivery approach. We can engender a burst for anti-inflammatory molecules or have a steady state elution profile for molecules for dry eye or glaucoma. This is truly a non-invasive approach. It's a steady elution and very efficient elution process, unlike eye drops where you have to overcome tear turnover. Our device is comfortable, has excellent retention rates, but it's cosmetically invisible, yet it's easy for the patient to identify, preservative-free system and favorable cost of goods and strong reimbursement. So thank you very much for your time.